Hello everyone, my name is Jacob Gator, and today I'll be presenting a paper titled Personalized Microbiome-Driven Effects of Non-Nutritive Sweeteners on Human Glucotolerance, which was published in Cell back in 2022. This paper investigates how non-nutritive sweeteners, or NNS for short, which are commonly used as sugar substitutes, affect the human glucose metabolism, focusing on how individual microbiomes mediate these effects. I chose this paper because I used to be an avid soda drinker. Some people like it for the combination, others use it for a source of caffeine. I simply just enjoyed the taste. However, as I'm getting older, I'm slowly starting to become a little bit more health conscientious, so I want to eliminate empty calories and decrease my sugar consumption, which got me looking into a plethora of alternatives. Obviously, drinking water is the best option, but variety is the spice of life and moderation is key. Let us first begin by stating our bluff, our bottom line upfront statement. This paper is a study in which the authors hypothesized that non-nutritive sweeteners influence glucotolerance in people in a microbiome-dependent manner. Another way to phrase this would be to say the microbiome composition of an individual plays a crucial role in how they respond to these non-nutritive sweeteners. This approach challenges our traditional all-or-one, it's good, it's bad way of thinking on this topic, suggesting a more personalized approach based on people's gut microbial diversity. I personally think it's important because people are actively trying to be healthier, and we need to be able to give them the right advice or information to make this decision properly. And information is constantly evolving and changing, so we need to stay up with the times. I've included their graphical abstract, which is the figure to the left. I personally like the design and think it perfectly summarizes the main takeaways of the paper. If you look at the bottom of the bluff, I've also included some famous examples of brands which utilize non-nutritive sweeteners. Again, it should be important to note that these things are famous examples. Non-nutritive sweeteners are in a lot more products than people realize. After this presentation, I implore you to investigate your pantries and see if any of your food products contain any of these non-nutritive sweeteners. I myself am going on a somewhat journey to find a good replacement for soda to decrease my sugar consumption, which is kind of, again, why I chose this article. So to list out the examples I have below, in pink we have Sweet and Low, which is those pink sugar packets found in breakfast places. In orange, I have included Mio, which is kind of like a juice concentrated that is added to water to give flavor. In green, I have Zevia, which is kind of marketed as being a healthy soda because it uses stevia extract, which is a natural sweetener. A more modern famous example of this would probably be Poppy or Olipop. And lastly, in blue, I have Diet Coke for my Diet Coke girlies. Moving on to the background of this paper, there is a growing interest in non-nutritive sweeteners, which are being used as sugar substitutes to reduce the calorie intake in food and beverages while still allowing them to maintain their sweet flavors. I would say this is due to us seeing an increase in the prevalence of health outcomes such as obesity and high blood sugar or hyperglycemia, and its linkages to diabetes and cardiovascular disease. These non-nutritive sweeteners like saccharin, sucrose, stevia, and aspartame are often promoted as being the safe, low-calorie alternatives to sugar. I remember when I was beginning my journey of looking for a better alternative to soda, people always recommended me Zevia because, again, it uses stevia extract, and people often conflate the fact that it's natural, so it should be healthier. Now, we can't just swap something within our diet and not expect it to have some kind of impact on our health or microbiome. As we continue to learn more and more about these sweeteners, it starts to dawn on us that maybe we don't have a consensus on thinking these sweeteners are benign. The authors talk about how there is this uncertainty in the efficacy of these types of randomized control trials. Some papers report improvements in metabolomic markers in response to non-nutritive sweeteners. Meanwhile, other randomized control trials report neither detrimental nor beneficial effects, which raises the concern over the long-term use of these products. However, the authors believe this variability and confusion between trials can be resolved by focusing on the impact of the gut microbiome. Lastly, the authors say that the other studies had two major limitations. Firstly, their ability to accurately screen and exclude individuals who have already incorporated non-nutritive sweeteners into their diets. And two, that their microbiomes were analyzed through 16S RNA gene sequencing or qPCR targeting specific microbes, which may not provide enough detail to assess the functional and species level impact of non-nutritive sweeteners on the gut microbiome. Now let's take a deep dive into the methodology of the study. It's going to seem like a lot, but I'm going to try to keep it to the main points of the paper. So between the years 2018 and 2020, the author screened 1,375 individuals for possible eligibility into the study. They want the study to only consist of participants defined as complete non-nutritive sweetener abstainers, meaning no one that consumed a non-nutritive sweetener, using a stringent screening protocol consisting of a detailed food frequency questionnaire that narrowed down that number to 120 participants. It's important to note a lot of people are not aware of how prevalent these sweeteners are in certain products. They had to turn people away because they unknowingly consumed something with NNS. They then divided the participants into groups of 20 participants for each of the 4 NNS intervention arms: aspartame, saccharin, sucralose, and stevia. All NNS were given as commercially available sachets containing glucose as a bulking agent. 2 sachets 3 times a day, corresponding to 8%, 20%, 34%, 75% of the acceptable daily intake, or ADI, of each of the supplemented NSS. Per US FDA guidelines, see reference 2. To clarify, sachets are those packets of sugar you see in coffee shops, meaning 3 times a day they drink water mixed with 2 sachets. Additionally, just to make sure we're all on the same page, let's go over the groups. The stevia group consisted of 20 participants, meaning 5 participants were administered stevia at 8% ADI, 5 participants were administered stevia at 20% ADI, 5 participants were administered stevia at 34% ADI, and 5 participants were administered stevia at 75% ADI. They also had 2 control groups. Since they used glucose as a bulking agent in delivering the NSS, they wanted to at least test that by itself, which is why you see a group labeled glucose vehicle. The other control group consists of participants who received no NNS, hence the name no supplemental control. The study involved 3 phases: baseline measurements of metabolic and microbial parameters for 7 days, exposure to nutritional interventions for 14 days, and follow-up for 7 days. Participants wore a continuous glucose monitor through the 29-day trial and underwent glucose tolerance tests for predetermined days. Anthropometric and blood tests were conducted on days 0, 14, and 28. Microbiome samples were collected from stool and oral cavity at predetermined time points. Participants logged real-time food intake and physical activity using a smartphone application. You can see this data outlined in the figure to the left, the black dots denoting the sample collection days. Since this paper is looking at personalized microbiome differences, they colonized adult germ-free mice with microbiota collected from stool samples via oral gavage, collected either at the beginning of the trial, day 1, or at the last day of the exposure, day 21, from all NSS supplement and control groups. Basically, they took 4 from the top responders and 3 from the bottom responders from each group and did a fecal transplant into individual mice to compare the mice glucose metabolic response to these new microbiomes. Let us now begin to go over the results. Firstly, you will notice these are big, comprehensive figures, so for ease of understanding, I have broken them into smaller boxes that I have denoted with a colored box to focus our attention when discussing them. Researchers conducted oral glucose tolerance tests, or GTTs, before, during, and after people were exposed to different non-nutritive sweeteners. They measured how people's blood sugar levels changed after drinking the sweeteners, using 2 differential statistical models to compare the blood sugar levels of the NSS groups to the control groups. You will see these models in the green box. Model A included the blood sugar level baseline and during time point, while the Model B included all time points, meaning baseline, during, and follow-up. They were able to measure this data using a continuous glucose monitor, which recorded intestinal glucose every 15 minutes. Afterwards, they calculated glucose tolerance test incremental area under the curve, or GTT-IAUC, to minimize the impact of variations in baseline levels when analyzing the data. Essentially, they normalized each person's blood sugar level to their baseline level and measured the incremental increase in glucose, and then compared the results across the groups during the first and second weeks of exposure and after exposure ended. They represented each person's data as dots with horizontal lines showing the quartiles. In both during 1 and 2 time points, we see a significant increase for both the saccharin and sucralose groups. Next, they averaged the blood sugar level for each person in each week of the trial and compared them to the baseline measurements. These are the figures in the red box. They present the results with black lines connecting the means, horizontal lines showing the median, and whiskers showing the 10 and 90th percentile. They used repeated measure two-way ANOVA and Dunnett tests to determine if there were any significant differences between the groups. They mark the p-values for the significance. So if you see if you see one asterisk, it has a p-value less than 0.05, and if you have two asterisks, it's a p-value that's less than 0.01. Again, we are seeing a significant increase for both the saccharin and sucralose groups, meaning glucose is building up in association with those NSS and has us believing it is impairing the body's ability to dispose of this extra glucose, essentially hampering the glucose tolerance. Researchers collected stool samples at regular intervals throughout the trial to study the bacteria in the gut. They performed shotgun metagenomic sequencing and used different software tools to analyze the bacteria's composition and how they work. For composition, they used Kraken-2 and Bracken. For function, they used the Metapsych, key models, and pathways. To see how the microbiome changed over time as the different sweeteners were added, the researchers used a special method called trajectory analysis. These figures are in the green box. They created ordination plots using tensor component analysis with M-product, or TCAM. This method checked if each sweetener group had a different microbiome path compared to the group without any supplement. TCAM looked at the change in each person's microbiome from their baseline. This allowed them to compare the changes over time. The results are shown in different panels, each corresponding to their associated non-nutritive sweetener. The data is shown with a mean value and standard error of mean, or SEM, and some comparisons were done using log2 transformations. To clarify the point a little further, Metapsych was used to look at the functions of bacteria in the microbiome, meaning how they break down and process different substances, such as non-nutritive sweeteners. Then, using the trajectory analysis, they track changes over time, meaning the closer the dots are on the graph, the more similar the bacteria are in terms of their function. If the dots are far apart, it means the bacteria are functionally different from each other. For these figures, we see that sucralose and saccharin are having a significant effect on the microbiome composition, while all intervention arms, sucralose, saccharin, stevia, and aspartame, are having a significant effect on the microbiome functions, the mildest being aspartame. Sadly, for the figures in the red box, we are seeing the top loadings for each Metapsych composition within each group, with hypothesis testing for trajectory analysis using the PERNOVA test. When the study talks about top loading, it's referring to the feature or data points that show the greatest variation or association within the groups being compared. In the context of the study, if there is an increase in certain bacteria or metabolic pathway associated with a specific function, like say a specific group of bacteria thriving due to non-nutritive sweeteners causing an increase in select pathways, and this increase shows a strong relationship with a treatment group, then this would be considered a top loading. The top loadings for each intervention arm are as follows. Sucralose is purine synthesis, saccharin is glycolysis and glucose degradation, aspartame is polyamine metabolism, and stevia is fatty acid biosynthesis. These results suggest that dietary supplementation with non-nutritive sweeteners can impact the functional potential of the human microbiome, meaning modifying the gut microbiome to fit its functional needs. The most prominent effect observed on the fecal microbiome observed in the saccharin group. Similar impacts were observed for the oral microbiome, changes in the relative abundance of select bacteria. In the sucralose group, there was changes in the relative abundance of 6 streptococcus species. In the saccharin group, the relative abundance of Fusobacterium decreased. In the aspartame group, the abundance of Porphyromonas and Provotolia nascininus decreased. So, in the first two figures, we saw an impairment in glucotolerance due to the buildup of glucose and functional modifications occurring in the gut microbiome. The next idea was to link these ideas together by identifying microbiome features that potentially contribute to this effect on glycemic control and how these alterations affect the host. They initially focused on sucralose because of previous data, but also because it's poorly absorbed and has a higher chance to interact with the intestinal microbiome. Additionally, it displays the greatest person-to-person heterogeneity, or difference, that could help them understand how the microbiome and metabolome continue to personalize glycemic responses. In the green box, we have a figure that is called a heat map. It serves as a potent tool for visualization of microweight data collected from NGS research. It will help us by providing a clear visual of how different microbial communities compare once supplemented with sucralose over the duration of the experiment. Now, in regard to sucralose, we see the purine biosynthesis pathway abundances were positively associated with the GTT-IAUC and decreased during the trial. Mixed acid fermentation in the TCA cycle inversely correlated with the GTT-IAUC, increased during exposure weeks, and trended toward the baseline during the follow-up. To read the graph, red serves as the row max, meaning a larger abundance. We are seeing an increase in those pathways. Blue is the opposite. It's the row min, meaning a lower abundance. Next, if we look at the box and whisker figures, we see 9 metabolites in the blood went up during sucralose, while 3 went down. No significant changes were seen in the blood metabolite after correction for the glucose vehicle and non-supplemental control groups. The TCA cycle pathway was more common in the microbiome, so the levels of isocitrate and transaconisate, which are part of the TCA cycle, went up in the blood during sucralose, while the levels of amino acids serine, N-acetylalanine, and aspartate, as well as aspartate metabolite quinolinate, also went up during sucralose. 2 more TCA cycle metabolites, citrate and fumarate, and several more amino acids, cysteine, lysine, and glycol L-valine, also went up during sucralose. On the other hand, in line with the reduction of microbial pathways linked to purine metabolism, the levels of pseudoaridine and uric acid in the blood went down during sucralose, as well as guanosine, 1-methylguanine, inosine, and paraxaxine. If you look at the figure in the red box, you will see they did a pathway enrichment analysis, which shows that amino acid metabolism and biosynthesis, as well as the TCA cycle pathway, were affected by sucrose impact on glycemic control. Lastly, let's look at the figure in the blue box. To find out which metabolite might be involved in how sucralose affects how well people tolerate it, they compared the metabolomic profiles of the top and bottom 5 sucralose responders. 3 metabolites were significantly different between the top and bottom responders. The ketone body beta-hydroxybutyrate, serine, and cysteine derivatives, cysteine, all 3 were lowest in top sucralose responders at baseline and increased during supplementation. None of these metabolites were significantly different between the 5 top and bottom responders in the glucose and non-supplemental control group. So this slide is going to be quite similar to the previous one. We are looking at all the other intervention arms in the experiment. In a parallel analysis to the sucrose, the authors also correlated the abundance of baseline metagenomic features with the GTT IAUC values in the saccharin, stevia, and aspartame groups. Furthermore, they analyzed metabolomic profiles in the top 5 responders for each group. In the saccharin group, baseline levels of provoldicolpre and UMP biosynthesis were positively associated with the GTT-IUC and progressively increased during the exposure period. Conversely, baseline levels of Bacteroides xylem solvens were negatively associated with the GTT-IUC and increased during the exposure windows. Many of the pathways negatively correlated with GTT-IUC were related to glycolysis and glycan degradation. Next, in the stevia group, 2 Provoltoli species were positively associated with the GTT-IUC and reduced during exposure, while Bacteroides coprophilus, Parabacteroides golistini, and Lacnospira species, which were all positively associated with the GTT-IUC, increased during both exposure windows. Stevioside was readily detected in plasma samples of the stevia group, exclusively during supplementation. Levels of the amino acids serine and lysine increased stevia supplementation. Notably, 2 metabolites of arginine, ornithine, and citrulline also increased during exposure. Lastly, in the aspartame group, B. fragilis and B. acidifications were positively associated with GTT-IUC, while B. coprocola had an inverse correlation. Levels of kynurenine, a metabolite associated with diabetes, increased during aspartame consumption. Now, to determine if alterations in the human microbiome contribute to non-nutritive sweetener-induced hyperglycemia, adult germ-free mice were colonized with stool microbiomes from 4 NSS-supplemented and control groups. They transplanted microbiomes from 42 individuals, the 4 with the strongest and 3 with the weakest response from each group, into mice. Let's first begin with our top responders, in the green box. Mice transplanted with day 21 samples from the top responders in the saccharin and sucrose group exhibit a significantly higher glycemic response compared to mice receiving the baseline sample from the same individual. Consequently, there was a substantial group effect for both saccharin and sucralose. Moreover, the microbiome from the final day of exposure of the top responder to stevia and aspartame also elevated the glycemic response in recipient mice for 3 out of the 4 donors. This resulted in a significant group effect for both stevia and aspartame. It's also important to note that none of these day 21 stool microbiome samples from the top responders in the glucose vehicle or non-supplemental control group affected glycemic response. Next, let's move on to the bottom responders in the red box. Again, in the saccharin group, the microbiomes for the bottom 3 responders on the final day of exposure still elevated the glycemic response in the recipient germ-free mice. Elevated blood sugar levels were observed in the entire cohort. Notably, no significant effect on glycemic response were observed in the bottom responders among the other treatments, including sucralose, stevia, and aspartame, or the control groups, glucose vehicle or non-supplement control, implying that a personalized microbiome-mediated impact on glycemic response in the transplanted mice were observed. These responses mirrored those of the non-nutritive sweetener supplemented donors, implying a causal link between MSS-related microbiome modifications and the impaired glycemic control. Now, our final figure aims to illustrate personalized microbiome differences by comparing the microbiome configurations of germ-free mice transplanted from the top and bottom human sucralose responders at baseline and after 21 days of sucralose exposure. Panel A serves as a schematic representation of the microbiome transplantation process, with red color coding indicating top responders and blue color coding indicating bottom responders. Panel B represents the principal component analysis, or PCA, of microbiome composition at day 21, revealing a significant difference between the two groups, which align with the human glycemic response data. Panel C, D, and E demonstrate that the glucose tolerance test, GTT-IAUC, exhibited similar results between the two groups. However, it is noteworthy that the top and bottom responders exhibited distinct microbial features at baseline, including species composition and functional pathways. These distinct microbial features may influence metabolic responses. Further analysis reveals 25 species and 27 pathways that exhibit significant differences at baseline. Notably, pathways such as menaquinol biosynthesis and purine biosynthesis were more abundant in top responders. Both of these pathways are linked to type 2 diabetes, see reference 4. A strong correlation between baseline glycolysis pathway abundance and glucose tolerance suggests its predictive potential. After 21 days, top responders exhibited higher microbiome diversity compared to both their baseline and bottom responders. Specifically, pathways related to fatty acid biosynthesis became less abundant, and changes in sucrose and glycogen degradation pathways were associated with enhanced glycemic response in top responders. These findings emphasize the significance of baseline microbiome configuration in shaping individual responses to sucralose exposure. I just wanted to reiterate our bluff and provide a quick little rundown on how they attempted to answer this question. Again, this is a study on non-nutritive sweeteners' effects on metabolism, and microbiomes reveal personalized and microbiome-dependent glycemic response. Firstly, researchers gave non-nutritive sweeteners to human participants and observed that saccharin and sucrose impaired glucose tolerance because we're seeing an increase in glucose. They discovered that these sweeteners were modulating the gut microbiome, leading to alterations in composition and function. They wanted to see what microbial features and plasma metabolites correlated with the non-nutritive sweeteners' effect on glycemic response. Thirdly, they wanted to confirm the microbiome's role by performing fecal transplants into germ-free mice and found that the mice developed similar impairments in glucose tolerance, which helped confirm that the microbiome is driving these effects. And lastly, it's personalized because each mouse received one participant's microbiome. This was so we could compare the top and bottom responders of the study 1-to-1. Like every study, there's going to be some limitations, and I just wanted to mention some of the limitations that both the authors and I think of. So firstly, they only included healthy, not overweight in non-glycemic individuals, which might not accurately represent the general populace. Moreover, the effect of non-nutritive sweeteners may differ between healthy individuals and those with cardiometabolic diseases. Secondly, the non-nutritive sweeteners used in the study were administered in commercially available sachets that contained a combination of glucose and the specific non-nutritive sweetener. It's possible that the microbiome and glycemic response may differ when the sweetener is administered with a commercially available sachet that contains something like carbohydrates as compared to glucose. And lastly, a longer exposure period to pure non-nutritive sweeteners may negatively impact metabolic health, suggesting further research is needed. Future studies should explore lower doses of non-nutritive sweeteners to determine their effect on the microbiome and host glycemic responses. And lastly, just some questions to consider following the end of this presentation. What specific metabolites are produced by the gut microbiome when consuming non-nutritive sweeteners, and how they contribute to glucose metabolism? How do these influence the host's physiology? How do different doses and exposure periods to non-nutritive sweeteners affect microbiome metabolic health? Is there a threshold at which we know it becomes detrimental to our health? What's the causal link between microbiome changes from NSS consumption and glycemic control and insulin sensitivity? Can microbiome-targeted interventions like, say, probiotics or prebiotics avoid the negative metabolic effect of NSS consumption, like impaired glucose tolerance? And lastly, how do NSS-induced gut microbiome alterations interact with lifestyle changes such as exercise, sleep, and stress affect metabolic health? Maybe even impacts like chronic inflammation. Lastly, here's a list of the references that helped me make this presentation. If you have a reference-specific question, just let me know, and I will try to answer it to the best of my ability. Thank you so much for this presentation, and I hope you learned something insightful about non-nutritive sweeteners.